welcome to our first webinar of 2022 for the Knowledge Portal Network. I'm happy to see all you joining us. For programming notes, again, this is a webinar, but today we're trying something a little different. So we want to engage interaction from you all. So if you have questions in the chat, please pop them there. Mackenzie's going to be monitoring them, and we're actually going to, if there's present questions, we're going to have us interrupt through Maria's demo today and actually have you ask them and have a sort of more of a dialogue than a polished presentation just from us and having you listen. So hopefully that'll be exciting. You can also put things in the Q&A as we go. This will be recorded as per usual and posted on the website for you to take a look at later if you want to. And again, I'm going to start off with a sort of a typical update on what's new and what's to come for the Knowledge Portal Network and the AMP CMD Knowledge Portal. And then today we're going to try a slight departure what we typically do at our webinars. Typically our webinars are about production releases and features that are on there fully integrated into the portals for your query. But today we're going to try something a little different and share an experimental feature because the goal of this is to engage and solicit your engagement. Our hope is that you'll see today's demo as a bit of a starting place for you to provide us with some feedback on what would it take to make this feature that you're about to see useful to you in your work, ideally daily. So we're hoping today is a little bit more of a spirited discussion and will ensue follow-up conversations. This feature is not available yet on the portal, will be in March, but we will gladly share with you a beta version of it if you're interested after this call to play with in a link that you can use yourself. So please feel free to do that. We're hoping that you will do that. But today you're going to get a demo of that feature in live time. So I want to start with the typical sort of updates of what's to come. One thing that's always nice is we have new members of our team growing every day. We welcome a new member of our team since our last webinar, Patrick Smadbeck, to our team. He's a computational mixed with an engineer. He has a little bit of both background, which is great because he's going to be working on implementing new and novel methods into the Knowledge Portal so that we can present more results from the data we aggregate. So we're welcoming him to the team. Other news and notables across the resource. The CMGGA just had a really excellent release this month of new data, particularly data relevant to CMD traits coming out of Bing Ren's lab, some chromatin accessibility from single cells for over 222 human tissues are accessible now on the CMDGA to you, along with a host of other QTL studies for type 1 diabetes. If you're interested, please go look at the website and see what they have on the CMDGA. And again, these results are going to be integrated in the CMDKP. You're going to see some of the first expression of that later today. Other features coming on board in March, we're going to have a production release. We haven't had one in a while because we've been really thinking about, as we've expanded to CMD, common metabolic disease, in the last year or so, we've been thinking about what are the core diseases we really want to feature in the portal across that space. So for that, in the next release, you'll see the largest meta-analysis for chronic kidney disease from CKD Gen coming online. It's already been published, but we fully integrated in the portal along with all of the other 300 or so traits you can peruse. We're adding another core CMD disease, NAFLD, to the portal. It hasn't been on the portal before, so those data sets are coming in as well. And increasing complications for type 2 diabetes and related traits, including peripheral artery disease and other associated complications. And in talking to our collaborators and our folks at the NIDDK, we really felt that we'd like to expand also into gestational diabetes and represent those results on the Knowledge Portal. So that's for the common metabolic disease space, but in other common disease areas, you'll see the additions of effector gene lists for osteoarthritis in the musculoskeletal portal. You'll also see the updated host genetic data for COVID-19 in the lung genetics portal. I think that's a really kind of exciting feature that we'll be adding to that portal in March. So look for those data sets coming online. On to features. So for features in March, one of the features that you may or may not have used on the portal historically is one of my favorite is sort of the first of its kind that we know of is interactive analysis of exomes. And we have this for 52,000 exomes for diabetes and related traits, along with the subsetted exomes from TopMed for interactive analysis. But what we're really hoping to do is expand that to whole genome data and allow you to do interactive non-coding burden tests across TopMed. This is a very new feature that we're hoping that will be the beginning of many data sets that will unlock many data sets in this space for you to do interactive analysis. And so this will require us to integrate annotations and different types of filters that you can pick and choose to run your burden test on whole genome data. So look for that in the end of March, and we'll have a whole webinar devoted to that feature, because that ought to be pretty dense new feature on the portal. And the other sort of branch of the knowledge portal space is a other platform we call Beor, or Bring Your Own Results. And at the end of March, we'll be soft launching this. We talked about this in our December webinar, where we talked about a space where we have the knowledge portals, but in many cases, researchers were coming to us saying, I have my results. We want to make them interactive and accessible, particularly for a manuscript or for the world to digest, because they're basically just living statically now, and we want to make them interactive. And so we've developed an entire platform around building a way for researchers to bring your own data and represent them in your own way. And this is an offshoot of the knowledge portal. What's great about this is it can leverage some of the tools in the knowledge portal, visualizations, and integrate with the knowledge portal more broadly. So to remind you of what the platform really, the hallmarks of that platform is, it allows you to bring your results the way you intend, represented your way, but make them interactive through a set of core capabilities, sort of features, plots, and illustrations that you can customize to represent your results. We collaborate to construct those, or if you want to do it yourself, you can. They're super easy, even as a non-computational folk. You can actually upload your own data and see them visualized in a very easy user interface. You can also stage access if you want to make it accessible to only you and a colleague, or you and a journal for review, or to the world. It's easy to do that, and there's levels of access that we build in easily to the resource. And finally, it's all a matter of leverage. For this, what we hope to allow you to do is take your results, and you can also see them with the integrated resource that we built for the knowledge portal, which includes all these genomic annotations from CMDGA, all the genetic associations across 300 and some odd traits that you can marry with your own data and present in visualizations we've created for you. So look for that. It's on the portal right now. If you want to actually, if you're interested in building a portal with us, please call us. We'd love to do that with you. So that's sort of the news and notables. But the most important thing we're here to talk about today is a preview of an experimental feature. We're calling this the Variant Sifter, sort of riffing on the gem or mining analogy I have in the portal, allowing you to explore genetic and genomic data. So I'm going to turn this over to Maria, but then I'm going to first sort of set you up in terms of how we visualize and envision this feature being used. So it's on the portal. It's eventually will be on the portal, but right now the portal is this integrated product that allows visualizations of genetic data sets, genomic annotations, and a host of methods. And it presents it to you as, if you want to search your favorite gene or region, you see your favorite region. And particularly right now here, I'm showing a region associated with chronic kidney disease, which happens to be very interesting because it's associated with a set of traits that, we presented sort of this distilled genetic summary of the genome-wide associations. And then you have your table of the lead variants for those associations. And then you have the elegant locus zoom presentation, which allows you to visualize the region, right? So this is sort of distilled visualizations allow you to sort of get a snapshot of a summary of what's going on in a given region. And in that same framework, you get a sense of what are the variants that are encompassed in that region. But really, you as a researcher who wants to prioritize a gene or a variant from a given locus want to select which variants may be most important to study, right? And so our goal is to create a feature that allows you to do that in a customizable way. So the feature that Maria is going to show to you allows you to take variants that are in your favorite region and filter them by a set of annotations, credible sets, associations, and results of bioinformatic methods that allow you to produce your own set of variants that you may want to select for perturbational screening or for downstream functional analysis. And this is the first of this feature, and we hope that this will be sort of expanded in many ways. One, in the types of data we represent, so working with CMDGA and working with our collaborators to bring in more traits and more genetic data to bolster it, but also to make the feature richer, to present to you actual information that will be useful in making decisions on which gene or variant to prioritize. So with that, I'll turn it over to Maria, who's going to walk you through a demo of the actual resource.
All right. We share. Okay. So yes, as Noelle said, this feature is going to let you really interact with the data, the associations, the epigenomic annotations, the credible sets, to prioritize what is of most interest to you. So let me give you an idea of how you will find this feature once we implement it in the production site. So right now, if you type a gene name into this entry box, it takes you to a region page, as Noel just showed, that's centered on that gene. And the genomic region miner down here gives you an overview of the associations. You can also add annotations and credible sets here. And there's a table of the variants in the region. So we're thinking that this view gives you kind of initial view of what's going on in the region. And then if you want to start really filtering and exploring, that you would head over to the Variant Sifter. And there will be a link somewhere around here that will take you to the same region and phenotype that you were looking at in the Variant Sifter. So here is the Variant Sifter site, and I'm going to illustrate just how to get around it. It's pretty complicated, but it needs to be in order to let you play with all of these different types of data. So I'm going to go to the same region that we were looking at. I'm going to put in a phenotype here. Let's do type 2 diabetes. And then I happen to know that this is the region. This is the same coordinates that were shown in that region page that we just looked at. So when you first see the page, you see an association plot. And this, okay, so we're looking at T2D associations. And we can see, as we saw in GEM, there is one index variant that really stands out here. And we'll be talking more about this variant later. So in addition to the association plot across the region, there's also an LD plot that shows the positions of, well, the associations and the LD relationships of all of the variants across the region to the index variant. And you can change the index variant in the display, and then you'll see the relationships of that variant with the other ones. So when we did this query, we got a table of variants, and there are 2,600 of them. So these are all the variants across the region that are associated with T2D with a p-value of less than 1. So this is a very wide net that we're casting at first. And you can sort this table by different criteria. So here we've got descending or ascending order of p-value, and you can see that this is the index variant that we've been looking at. That's the most highly associated. Okay, so we've got this large set of variants that are associated with T2D, and now we want to see what else is going on with these variants. So we have the ability to filter by credible sets. So as I'm sure you all know, a credible set is a group of variants that is predicted by, and there are various methods to do it, and some of them take into account, prior biological evidence. Some are more computational, but there are a set of variants that are predicted with a certain probability, usually 95% probability of including the causal variant in the set. So right now we have two different credible sets, two different sets of credible sets available. And of course, we'll be putting more user-friendly names for them in this interface. But one of the sets is from the Diamante European study. So this was a large study published by Mahajan et al. Of T2D GWAS. And when we can select that credible set, so actually it only has one member, and it is in fact the same index variant. And you can see that when you select a credible set, the table is immediately filtered down to just the members of that credible set. We have another credible set, another set of credible sets available. These were calculated in Hilary Finucane's lab for 40 different phenotypes using UK Biobank data. So we can add that credible set to the display. This is a bigger credible set, although it includes the same index variant. And now the table contains all the variants that are in either of those credible sets. So this is a good way of filtering down very quickly to a group of variants that is most likely to include the causal variant for the phenotype you're interested in. So now I'm going to get rid of these credible sets to get us back to our initial large set of variants, because I want to illustrate another type of filtering. So in addition, you can filter by tissues and annotations. So I think you're all aware that, and as Noel mentioned before, our sister resource, the CMDGA, aggregates and processes epigenomic annotations that reflect the regulatory potential of different regions across the genome. And so they transfer these data sets to us, and we run the Gregor algorithm, which looks for enrichment. It looks for enrichment of genetic association signals for a given phenotype within an epigenomically annotated region in specific tissues. So this is a good way of suggesting which tissues might be most relevant for a particular disease or trait. So when you when you add these tracks to the Variant Sifter, there's a large selection of tissue groups. These are kind of broad groupings of tissue, and within these there are much more granular tissue types and cell types that belong to these groups. But our colleagues at CMDGA have narrowed it, have categorized them in this way so that it's easier to navigate this list. So you can select from all these tissues, and you can also select an annotation type. And right now we have these 4 types of annotations, but we're working with our CMDGA colleagues to add more. I think the next ones that we'll add are going to be target gene predictions, for example, from chromatin conformation studies, and also QTLs of various kinds. So that's a lot. There's several different annotation types. There are lots of tissues. How do you know what's going to be the most interesting to look at? Well, this global enrichment graph is meant to show you that. So this graph shows the enrichment using Gregor of genetic associations for T2D across this region. And so Gregor, the Gregor output is both a p-value, which shows significance, and a fold enrichment. And so this plots both of those things. And these dots are color-coded by annotation type and also indicated by tissue here. And the most significant tissues are spelled out in the graph, but you can also mouse over the other ones to see what they are. So this graph gives you, at a glance, an idea of what might be most interesting to look at in this region. And pancreas certainly stands out with very significant enrichment and a good fold enrichment too. So, and this is a blue dot, so we see that it's an enhancer. So let's first select enhancer. And what we see now is plots of the enhancers across the region in different tissue types. So this shows a lot of tissue types. These are the ones that have the most significant enrichment for enhancers in the region. Excuse me. And now, since we want to focus on pancreas, let's add pancreas. And so this track shows the locations of enhancers in the pancreas. And here we have the enrichment values for pancreas and enhancers. And these are the same values that are shown in this plot. So what you see immediately is that we started with 2,600 variants. Now we're narrowed down to 391. So now the table shows just the variants that are significantly associated with T2D and are in regions that are annotated as enhancers in pancreatic tissue. So, so this now gives you a much smaller set to work with. And this is very versatile. You can also add filter and add more tracks. So let's add binding sites, for example. Now we see a track that shows binding sites in the various tissues. Now the table is filtered even further. We have just 56 rows. These are all the annotations that are associated, all the variants that are associated with T2D and are in pancreatic enhancers and are in pancreatic binding sites. So we recognize that people are going to want to filter these data in many different ways. So you might not just want to do find variants that answer all criteria. You might want to add to make an or statement, are they an enhancer or a binding site? And there are lots of different combinations. So that will definitely be a functionality that we're working to add to this interface. Okay, so Yes, and I just wanted to point out that we still see our index variant here, and I cheated a little bit because I already knew from the literature that this index variant, this was studied by Kyle Galton and colleagues. We know that it is in fact in a binding site for the FOXA2 transcription factor in the pancreas, and it's an enhancer also, and it's this is the causal variant for this, well, very likely the causal variant for this region. So, but it definitely stands out and comes up in this analysis as the most interesting one to look at in this region. So, oh yes, and I also wanted to mention that you can export this, what's in this table, as a JSON object. And then you can take that object and plug it right into our Bring Your Own Research framework to create your own tables or visualizations. So that's a handy way to take these results and work with them further on your own. So there's even more versatility to this interface. We're just looking at one phenotype right now, but actually you can add more. So let's add another phenotype in this region. Let's do fasting glucose. And when you do this, you have several choices. If you say new search, that will just get rid of the glucose, excuse me, that will get rid of the T2D associations we've been looking at, and it will replace it with fasting glucose. But then you can either choose overlapping. This will show us the variants that are associated with both T2D and fasting glucose, or you can look at all variants, which gives you all the variants associated with either phenotype. But let's look at only overlapping. So now, and I hope you're noticing how fast this interface is. I love it. It's a pleasure to play with an interface that's so responsive. And I should also mention that this is all the work of my colleague DK Jang, who has designed all these visualizations, and it really works great. So, okay, now we've got a plot for fasting glucose, and now we have a combined plot that shows the variants that are associated with both T2D and fasting glucose, and the variants are connected by an arrow, and they're color coded. So for example, these are the green are the fasting glucose associations, and these variants are connected by a vertical line with the blue associations that are the T2D associations for that variant. Okay, now that we've added a second phenotype, the tables change too. So since we're looking at only the combined, only the overlapping annotations, now we are seeing only those, variants, not annotations. Now we see only those variants that are associated with both T2D and fasting glucose, and they're in a pancreatic enhancer and binding site. So you can see the potential to keep filtering this down and doing different things with it. So let's see. Yes, I should mention that the global enrichment, when you add a phenotype, you get a new graph that shows the enrichment for that phenotype, because the enrichment is obviously phenotype specific. You get new LD plots. So, and you can keep adding phenotypes. I'm not going to do that because it's going to get ridiculously complicated, but you can add as many phenotypes as you want and stack them up. You can also zoom and pan. And yes, you can filter if you just want to see one particular variant or have a p-value cutoff. You can filter using these boxes too. So that was quicker than I thought, but I think that is pretty much the functionality of this interface at the moment. And I do want to emphasize that we really want to work with you on this, and we'd like you to think about how you could use this and what types of features would make it more useful. And it would be great to just develop this in concert with the community. We'd like to know what else would make this interface even more useful. What features would you like to add? So this didn't take very long, but I think I'm going to stop and hopefully people will have questions, because I'd love to make this a little bit of an interactive session.
One thing to note that I think Maria mentioned, but is that right now one of the features, that major caveat that we know that we want to add in the release in March is right now it's all the union. So it requires that all of them, the variant filters to must be, associated with this, associated with that, overlap with this annotation, be in a credible set. But what you may want is some and or options, like if it's in pancreas or, my other favorite tissue and so on and so forth. So we plan to allow different filtering based on and or union combinations, because I think that's one of the limiting factors right now. But yeah, as Maria said, one of the things about this, that's the reason that we're putting it out there, is because it is essentially a framework by which we would add the right types of data types and filters that you would find useful in prioritizing variants. So I think that's the beginning of the conversation. So I'm seeing the questions coming in the chat right now. So let's go to those.
Yeah, I see that Jennifer asks, how can you go back and show us how to get to the sifter? Well, there is no link to it at the moment because this is just a beta development version. However, we can send you the link if you'd like to play with it. And after our March release, you will be able to get to it either from the region page or from the KP Labs, Knowledge Portal Labs page, the menu.
And then Jen also asked, will there be more details on binding sites if you scroll over them? Ultimately, yes, it's a really good point, Jen. I think that's one of the biggest challenges as we figure out how to unravel the connections between, the collapsed track and the annotation in CMDGA. As a first approximation, I think we're planning to at least allow you to get to the source of that data in CMDGA through accession numbers. That's what links them. I think that's the first plan, and then the other one is to build a way to visualize them or give some sort of more detailed information. That's coming. But right now, it is very compressed, so you really don't know, what annotations go into that particular, track or that summary. And then one other note, right now, Gregor is the basis of our global enrichments. We're going to be updating that to LD score regression as well in the future for more annotations.
So
Yes,
we have a couple questions in the Q&A. So our first one is from Douglas, is in the annotation tracks, can the specific top variants or other variants be located along the chromosomal positions visually?
In the annotation track. So, well, you can, I'm not sure I understand the question. You can line them up. When you scroll over this, you don't see the variants. These are just the annotations, but I'm not sure I'm answering your question. Can we unmute?
Yeah.
Red one to Mackenzie. Can you do that?
Yep.
Yeah, I was, yeah, you can sort of scroll down and try to line them up, but it's where these features, these annotation features occur. I just want to make sure you can actually tell which variant is overlying some of these binding site features, other than just eyeballing it up vertically and scrolling back and forth. Is there a way of like having your top variants shown somewhere along the annotation tracks?
Mm-hmm. Or at least just maybe a crosshair would be helpful. That
Across there, maybe, yeah.
Down across these plots. Mm-hmm.
Yep.
Yeah, good point.
Highlighting them, I like that.
You do also have, for each variant, you've got the coordinates of the annotations that it overlaps. So that's one thing, but I agree with you. That would really help.
So our next question is, could the framework be used to load additional data sets? So essentially set up locally. It's probably in context of the T2D portal as well.
Yes, I think so. This framework, I think, can be used as part of the research portal framework. Although I don't think the research portal database can hold whole sets of summary statistics, but it can draw on our bioindex where these things live, or on other remote sources. And I think DK is on. If our designer, if you would like to comment more, DK. I'm not sure if he is on. Other questions?
I don't see him.
Yeah. Oh, DK says, can you unmute
DK
me?
should be unmuted. Hold on. I think I can do him.
I just gave him permission.
You hear me?
Yes.
Yes.
Yeah, so as long as your data is formatted the way the framework can consume, then you can plug in any kind of data into it. So actually, this framework at this point, this framework is shared by two projects. One for the KP portals, and the other one is for other project. So it's just neutral, and then you can take any data formatted in the way it can consume. Yeah.
Thank you, DK. Are there any other questions from the participants?
Maybe not at this time. Okay, well, of course, you're always free to email us at help@kp4cd.org, and we can answer questions. But yeah, anyone who would like to use this interface, just play with it beforehand, please let us know. We'll give you the link. And 1 month from today, March 10th, at this time slot, we're going to have a focus group. So in that group, anyone is invited, and we're just hoping to have a really interactive discussion. So if people would look at and play with this interface beforehand and think hard about what would make it more useful to you. And then we'll have a really focused discussion about changes we can make to this interface and ways we can develop it. Yeah, and I think that's it. We'll post the recording of this later. And yeah, and Mackenzie, do you have any further parting thoughts? So I think
you wrapped that up well. Our next webinar will be April 14th at the same time. So please be on the lookout for information regarding that as well. Thank you all for attending, and please feel free to reach out to us if you have any questions.
Thanks, everyone. Bye-bye.
Bye.